My name is Paul Isaac, and I want to do a science fair project on DNA methylation in Chlamydomonas as a result of heat stress. So the idea for this project first came to me when I wanted to do a project involving allergies and basically factors that would affect that in children. That was when my advisor, Dr. Granger, pointed me out to the field of epigenetics, which basically proves that Lamarck's argument of organisms inheriting traits that their parents developed over a lifetime was not entirely baloney. So as I looked further into this and realized that it's pretty complicated to get human testing involved in a science fair project, we eventually came up with the idea of using algae, since they're quite present in ecosystems and that they've proven to be a problem with algal blooms, and they've definitely changed as eutrophication and global warming has occurred in this world. So we thought that they'd be a perfect organism to test epigenetics on in something relatively simple while also being relevant, such as global warming and then therefore heat stress in water. So basically, me and my advisor worked on this project for a long time, since January of last year, and since then we've been troubleshooting a lot of problems. Basically, I knew very little, if not nothing, about the field of epigenetics and all the genomic techniques involved in said field, but through the guidance of my biology teacher, Dr. Granger, my advisor, I was able to get a lot of the resources to accomplish my testing and also get a lot of literature to read up on to understand what I was doing. We found a couple model studies that we based our trials off of and where to go, where to get our grow mix, and etc., etc. And basically, my advisor, Dr. Granger, pointed me in the direction of the things I needed, and whenever I had questions, she was glad to clarify upon them due to her background in the bioinformatics industry and biotechnology industry. So she really helped me in basically coordinating all the resources I need for the project and helping me greatly troubleshoot some problems. So basically, a couple of the key problems that we had in this project were, one, getting extraneous DNA or extraneous readings on our PCR data on our PCR products, which we solved through the purification process, which took a little additional investment monetarily, but we felt that we were getting more consistent results. Also, a problem arose when we had to decide which DNA sites that we would be testing for methylation, since methylation requires a special form of PCR called qPCR if we wanted to test for multiple gene sites. So we ended up testing for a couple heat shock proteins, and we also tested for a couple control genes in algae that were known to not change. And then based off of that, we've been able to extrapolate some data that can prove that heat stress might have an effect on epigenetic regulation in algae. Thank you.